There are bacteria which are thermotolerant. They can tolerate high temperatures. But then there are bacteria which are thermophiles, that they prefer high temperatures. The word thermophile literally means heat loving. And so thermophiles prefer to live at more than 40 degrees Celsius. There are even hyperthermophiles which can live at over 80 degrees Celsius, even over 100 degrees Celsius. Perhaps the highest is slightly over 110 degrees. How can they do this? Thermophiles and hyperthermophiles have a number of molecular modifications which allow them to live at high temperatures. So, for example, most hyperthermophiles are in the bacterial group of Archaea, and they possess ether bonds in their cell membrane lipids, and the cell membrane fatty acids can fuse. So instead of having a bilayer, they can form a monolayer where four lipids fuse together, forming a tetraether. This makes the cell membrane stronger and more capable of resisting high temperatures. The enzymes which work with DNA are unique in hyperthermophiles. So, for example, the topoisomerase and gyrase enzymes are unique, which control then the coiling of DNA, making it supercoiled in a positive direction as opposed to negative. And so this is an adaptation to high temperature living. Since heat can change the folding of proteins, the amino acid sequence of the proteins of hyperthermophiles are different because it then allows for a folding which is stable at a high temperature. This is actually quite useful in biotechnology. For example, in polymerase chain reaction, the DNA polymerase, which was extracted from bacteria adapted to very high temperature, is used to replicate DNA, say DNA from a crime scene, because then the sample can be heated to boiling and the enzyme from these hyperthermophilic bacteria remains stable. Thermophiles and hyperthermophiles can be found in a variety of environments today, such as regions of the seafloor with tectonic activity. So in the water and the sediment layers around tectonic activity, say at a mid-oceanic ridge, there is high temperature where you can find hyperthermophiles, which are mostly archaea, but which do include some eubacteria. There are thermophiles which have been isolated from coal deposits and petroleum deposits, which apparently are the descendants of the bacteria which were present in the organic matter when this original deposit was laid down before it was converted to coal or petroleum, and now being buried exists at a higher temperature. When igneous rock forms, there are reduced elements which could be used as food sources. And so once it cools sufficiently, thermophiles and hyperthermophiles can penetrate rock layers in the groundwater seeping through, which include lithotrophs, which can metabolize the inorganic matter to produce energy, and then other hyperthermophiles, which then live on sugars presumably produced by the lithotrophs. And so there can be a complex food web in these microbial populations. So once again, thermophiles and hyperthermophiles are a diverse group of microbes, some of which are metabolizing inorganic minerals, some of which can metabolize organic molecules, some do both. Some are anaerobic, living in deeper sediments, say around a volcano, while some are aerobic, living on the surface.